going here because everyone has these diverse different methods that we're using, and for instance I do neuroimaging and aphasia research, and I hope that there's a lot of people as well that are interested in that. So with that, I would like to welcome Brielle C. Stark. I made sure to use the C, which is important for the authorship. She got her PhD in clinical neuroscience at Cambridge, and she's now an assistant professor at Indiana University, and she's done a lot of work in neuroimaging and aphasia, and she is going to talk about a number of data sets and platforms that are online in terms of aphasia and other atypical populations. So with that, please welcome Bree.
Thanks, William. Can you hear me okay?
Yeah, you sound great.
Great. All right, let me share my screen if it wants to play nicely. All right. So Brian McWhinney had to step off for another call, but he hopes to jump back in at the end. So hopefully we'll get some insight from him as well. So Brian McWhinney is the person who created TalkBank and who gets all of the funding to keep it going. So I hope he jumps back on because they've created some really cool tools. So as William said, one of the things that I do is figure out how we can make clinical language data a bit bigger, how we can use bigger data to answer some of our core questions. I've always looked at this through the lens of aphasia as well as typical aging, so this talk will be a little bit more about that, but there's tons of other resources that I have on here too, so I'm hopeful if you are not just fully here for aphasia, you'll still find it interesting.
A quick question, Bree. Mind,
yeah.
I think you're showing your presenter view rather than the
oh, weird. Okay.
So it's kind of a little bit weird, so just in case.
How about, oh no, how about that?
That's much better. It still seems to be kind of strange a little bit on my screen, at least like it's kind of cut off on the left. I don't know what that is from, but maybe you can just show the slide. Yeah, that's better. That's good.
Okay.
Yeah,
I had two monitors plugged in, so it didn't like it.
Yeah, I've had
it. So now I have one monitor. Okay, so just to highlight, so talkbank.org is where all of these things currently live, and the goal of that is to bring together a whole bunch of resources from a variety of places and put it all in one place to help those of us who are interested in big data, specifically language-related data. This is what the website of TalkBank looks like. Hopefully Brian can jump in at the end because he wants to talk about this new thing that they've just instituted called TalkBank DB, which is essentially a really great way to search the databases without having to kind of go in and manually scroll. You can actually use search terms to figure out what databases to use, as well as some other really cool things. But TalkBank has 14 different research areas that it supports. They're all called banks. And I've highlighted them here. So there's some that are based on conversation, so more dialogue based. And those are CA Bank, Samtail Bank, and then Class Bank. They've got child specific ones, which you can see there as child language banks. Many people are familiar with Childs, which is also a part of the system. But there's also Fawn Bank and Home Bank. Some of those have conversations and some are more monologues. They have multilingual specific banks. You can see here a bilingual one, a second language one, and then they have clinical banks, which they continue to grow, which are invaluable resources because for those of you like me who collect clinical data, it's really quite difficult to find participants who fit your inclusion criteria. So having this available is fantastic. So I'll show you kind of what the things look like in a second. I'll talk a little bit more about AphasiaBank just because it's kind of the easiest to talk about since I use it the most. I do want to mention that even though there are multilingual banks specifically, a lot of these banks like AphasiaBank have, they collect demographics that discuss whether the speaker is monolingual, if they're bilingual. I don't know what proficiency metrics they use, but they do make it available, that type of demographic information available. And AphasiaBank is now available in something like 8 or 9 different languages, Spanish, Mandarin, Cantonese, etc. English is the biggest at the moment, but there are lots of different ones available. So it's not just English only. So in order to access it, it's all password protected. Each bank comes with what are called ground rules. So all you have to do is read those, and then you email either Brian, which his email's on here, or Davita Fromm if you're really interested in AphasiaBank, and her email is on the AphasiaBank main website, which I'll show you a little screenshot of in a second. Students can join. I make sure that if I have a PhD student or a master's student in my lab who's working on this data, I sponsor them as a faculty member, and then they join as a member if they're interested in using this. And they also have some really great things you can use in class too, if you want to use it as a teaching resource. So this is what the main page of AphasiaBank looks like. Let me see if my little pointer works here. Can you see my laser pointer?
Yes.
Oh, fantastic. So on all of these banks, they're set up in a very similar way. So right now, my little laser pointer is over AphasiaBank protocol. If you click on that, it will say exactly what data is collected, what tests are used. So for instance, in AphasiaBank, they have demographics that they collect, and you can see what demographics they're collecting at all of these sites. They do a neuropsychological battery, including a naming test. I believe it's the Boston naming test, really detailed repetition battery as well. And then they do a spoken discourse protocol, which is all monologue, it's not dialogue. That is based on picture descriptions, procedural descriptions, narratives like story retelling, personal life events, things like that. And for AphasiaBank, all of the spoken discourse stuff is videotaped and already transcribed for you. For reasons related to PHI and protection of health information, I'm not going to show you the whole interface because it would show an individual's face. So I'll show you in a second what a transcript looks like. But I do want to mention that the neuropsych battery, for instance, the naming tests, are not fully transcribed just yet. That might happen in the future, so I've been told. But one way you can do this on the AphasiaBank website is if you're just interested in browsing some of the data that's available, you would hit this browsable database. That will take you to some choices. If you're interested in speakers with aphasia, you would then click the aphasia link, and then it would take you to languages that you're interested, right? So if you're interested in English, you would continue there. And then it's organized by site. So for instance, Julius Fredrickson from University of South Carolina, he collected some data that's now on AphasiaBank. So you'd click the Fredrickson site, and it would take you to all of the participants that he collected at South Carolina. There's something like 20 sites at the moment that collect aphasia data, and you can always become one too. So just email Brian to join that. You just have to edit your IRB accordingly. They also have things that are called non-protocol. There's a lot of interesting things like group therapy sessions that are recorded. But since they're not part of this giant protocol, they're not transcribed. So you can still search those, you can transcribe them on your own, but they're not as easily accessible. So one of the things that I think AphasiaBank is fantastic for is the fact that it's already transcribed for you and coded. So it uses CLAN, ChatCLAN, which is a type of coding and analysis software that Brian and his team have come up with over, I guess, the last 20 years now. I think he published a manual on it in 2000. It's fantastic. It's all freeware. It's compatible with almost every operating system. I think they're working on Mac Catalina right now. And what you get is a transcript that you can download and then manipulate using the CLAN software. So right down here, this is what your CLAN interface window looks like. That's where you tell it the commands that you want. It's a little bit like coding, but it's a very basic thing to learn. The manuals are fantastic. They kind of lead you step by step, and there's some tutorials as well on the AphasiaBank website and TalkBank called screencasts that will walk you through these things. If I can zoom in, and hopefully this works, all of the transcripts are coded with gestures, pauses. They're coded at the word level as well as at the utterance level. They give you a fantastic amount of information about errors like paraphasias, morphological errors, agrammatic or paragrammatic sentence or utterance level. They give you a just fantastic amount of data that you can ask it to output later. And they're all checked on the transcripts. Quality encoding is all checked by the team at TalkBank. Just to give you an example of some of the information you can get, this is called the eval tool from CLAN. And it gives you really valuable information like speaking time. You can ask it to do it for all stories. So for instance, you're interested in the story retelling of the protocol, which happens to be Cinderella. You can ask for information for every speaker from Cinderella, or you can ask for every speaker across all stories, for instance. It'll give you things like mean length of utterance in words, or you can specify if you want it in morphemes. It'll give you total tokens, things like that, percentage of parts of speech. It gives you an enormous amount of data, and it's a fantastic way to probe language structure, but you could also use it to do language function, right? If you wanted to go in and code things related to story grammar, cohesion, coherence, main concept analysis, core lexicon, all of those you can use this data to do. They're not coded specifically for those more discourse level analyses, but they're free for use once you're kind of a member of this team. So then I thought, well, William suggested I talk about kind of how I've used this data as maybe to probe some insight or give some examples. So this is not AphasiaBank data, but this is the chat clan coding, just to give you an example of how this might be used. So William mentioned that I do have a degree in neuroscience, so I approach everything related to language from the point of view of kind of what's going on in the brain. So what we did with my colleagues at South Carolina is that we went in and we coded a whole bunch of picture description, spoken language from individuals who had had a left hemisphere stroke. And we coded a whole bunch of paraphasias or word level errors. And we looked at the brain damage or the lesions that were associated with semantically related errors versus phonemic errors, for instance. And having chat clan coded, doing our coding, making sure we were reliable, but using that system allowed us to get this information really, really easily. AphasiaBank, TalkBank do not come with brain data attached to them. I think that's a pie in the sky dream that Brian has. Maybe someday that will come to fruition, but you are able to probe more deeply into the behaviors from a very large data set, which is now what I thought I'd give some examples of. So how I've used AphasiaBank. I did publish an article most recently in American Journal of Speech Language Pathology. I think it was 2019, which you can see there on the left. And then my colleague Julia and I are trying to currently get this thing on the right published at some point soon. Cross your fingers for 2020. So let me give you a little insight into what I was doing there. So in the AJSLP paper, we were trying to figure out essentially, do speakers with and without aphasia show task-specific linguistic differences, as in linguistic microstructure? So we were really looking at some of these variables you see here, such as mean length of utterance, words per minute, verbs per minute, or verbs per utterance rather, type token ratio, those types of things, kind of singular measures of discourse that one might be interested in that tells you something about the quality of the structure of the language, very much kind of at that structure level, not so much functional level. But the cool thing we were able to do with AphasiaBank is we had an enormous, for aphasia, sample size. So we ended up having 90 individuals with aphasia. We got 84 age and education and sex matched controls, also from AphasiaBank. And we were able to really directly compare how individuals differed on these kind of language outcomes across tasks. So across narrative tasks versus expository tasks versus procedural tasks. What we wanted to do after that is expand a little bit and do a bit more of a sophisticated analysis, which is what Julie and I were working on for the past year or so. And this is again taking all of the data available in AphasiaBank, which is reaching close to 300 speakers with aphasia at the moment, over 200 controls of various ages. We wanted to again look at kind of linguistic structure by task, but rather than do singular metrics like mean length of utterance, right, which everyone kind of disagrees on what that truly represents, why not just compare all of this data in a multivariate way? So what we did is we took bunches of parts of speech, so all the part of speech data that you get from the CLAN output, tense usage, all these interesting structure variables, and we wanted to model them in a multidimensional space and then reduce down our space so we can kind of interpret it. And we wanted to look at the difference between tasks and, again, that linguistic structure. So on the left, you can see just a comparison of controls, whereas the yellow is a procedural task where people describe how to make a sandwich. Cinderella and important event there in the middle are both narrative tasks. One is autobiographical, tell me about your life or an important event, and one is a story retelling, which is the Cinderella story. And then you have expository tasks. One is called the cat rescue, which is a picture of a cat being rescued. And then window or broken window, which is a sequence of pictures that people are asked to describe. And we wanted to see if there was a difference by task, and you see it in for linguistic variables of interest. You see it for controls, and you also see it for speakers with aphasia. It's probably easier to look at this bottom graph here, which is just kind of narrowing down by aphasia type if you're interested in that, and seeing that there's some task similarity in terms of linguistic structure that breaks down there. We actually go into more detail and we look at severity as well. People with aphasia have various severities. So that's something we're also interested in kind of looking at. So that is how I have used AphasiaBank. Many publications are out there using AphasiaBank. Many of them are hosted on the AphasiaBank website. So you can get a great idea of all of the interesting ways to use this. But I didn't want to just give you things about AphasiaBank. I wanted to leave you with some other resources as well. So if you want to screenshot this is a great one. I'm sure this will be recorded for later. But Oscar is a fantastic resource. It's, I think, hosted at Northwestern. Lots of speech data available there. There's some resources for data collection like iPhod, which is English words and pseudo words. They've also got things like Clicks, which I believe is cross-linguistic, which there's at the bottom. But then there's also been some really fantastic things available in conversation. My personal favorite is the Carolina Conversations Collection. They have 2 cohorts of adults here. One cohort kind of talks about their chronic disease. Another cohort talks about their cognitive impairment. They rotate who they are speaking with. So if it's a student versus kind of a known peer, and they also have longitudinal data on some of those speakers, which is really unique. That's not typical. DementiaBank, which is another part of TalkBank. If you go to the PIT database, it's one of those kind of by site organizations within DementiaBank. They have a lot of longitudinal data on a picture description, a sentence generation task, I think word fluency as well for individuals who have various types of neurocognitive disorders, including mild cognitive impairment, Alzheimer's disease, and then I think they have a few primary progressive aphasia as well. And Johns Hopkins also has a database thanks to R.G. Hillis, which has some primary progressive aphasia also included on DementiaBank if you're interested. There's a really great blog I just left here on the bottom. It's a WordPress site. Hopefully it lives there forever, but if it doesn't, go copy it to a Word document somewhere. But it has fantastic resources for open source language, either data like AphasiaBank or things to use in your own data so you don't have to create your own things. And just a little plug before William makes me stop and answer questions. If you're interested in anything related to spoken language and aphasia specifically, please join this working group. We're kind of trying to improve the evidence and work together to make these databases even bigger. So that's my little plug, and I guess I'll take questions unless Brian, did Brian join back?
Sure, yeah, I'm here.
There he is.
Turn on my video.
All
Great,
right.
well, you know, it's great so I don't have to say all that.
Yeah, I tried to do it for you, and obviously you're the man, the leader. So
well, you know, once you get to be 74, we're very happy to have these young people who are going to take over pretty soon, I hope. So all these types of data, I think eventually we're going to have so many types of database like this that we're going to have, you know, lots of different input, not just aphasia, but a lot of it is child. I just want to add a few things. So first, for the non-clinical, there's no password required. So Childs, Biling Bank, so on, totally open. Also, you're obviously, your work depended a lot on the fact that the data were tagged morphologically. You didn't show that in the particular ones you showed, but you know, from all of AphasiaBank, all of the English trials, a lot of Spanish, German, French, we've automatically tagged by parts of speech and then by grammatical dependency. That isn't true. I think we have 12 languages we do that way. So that's an important thing. In terms of Dementia Bank, I think it's very interesting to note that there's now a challenge at Interspeech for the best computer program that will be able to differentiate mild cognitive impairment from normal, from really real dementia. And we have now something like 150 computer science labs around the world that are, you know, basically using these data to make the most wonderful algorithms they possibly can. So that's a very speech technology type of world. There are also speech technology things for aphasia that some people, particularly for apraxia, that, you know, people are getting into. And then within the child language area, there's a project called FonBank, which really looks at young children's phonological productions, and it has a program called Fon that has integrated within it all of Prot. So you can run Prot inside Fon and do all these fantastic, you know, pitch extraction and following jitter and vocal fry or whatever it is, or IPA or whatever you want to do in phonology. So I think those are important things. Let's see, one thing is, I don't know if I can share my screen here. Oh, I can't share screen, okay. But if you go to the TalkBank site, there's a link to a tutorial page that I think would be very helpful to people. In the tutorials are all screencasts for all these different you know, facilities inside
I think you plugged them.
TalkBank.
Those are great.
Yeah. And you know, not to push too well. So there's two other things. One is that we really do want to move more and more to linking to the brain, just like you're doing, Brie. And right now, I think the best approach would be to go to OpenNeuro. We're working with a project over at Pitt with Julie Fees that will be taking AphasiaBank data and also will get scans. We're mostly talking structural here. You know, it might be some functional, but mostly structural, and including white matter. And I think that is very important. So anyone who has ideas on how we should best do that, my idea would be that we could keep all the language data on TalkBank, but then link to OpenNeuro for specific data sets. So, you know, I think hopefully that would work, but we haven't really done that yet. And then the other is this TalkBank database, TalkBank DB. There's a link from the TalkBank page to TalkBank DB. Right now, it only goes to child's data, but really by the end of the week, we're going to be, we already have pulled in all the other databases. So AphasiaBank, Dementia Bank will all be searchable. In that system, you know, you basically specify what data you want, whether you want types or tokens. You can use CQL, which is Corpus Query Language, to look for sequencing of grammatical structures. And then you can get all the matches to those queries in a downloadable CSV, which you could, you know, then pull into R or into Excel or whatever. But also, we also have a package inside R where you can pull directly using R queries. So that I think is going to be a very important thing. We haven't got everything out there on the web, so I would say try it next week. Okay, so that's enough to add.
I think there's a few questions you can answer better than me. I'm just reading off of this.
Okay.
Yeah,
Are there any
questions?
Oh, I was just going to read off of this.
Oh, sure, yeah.
Are there any banks for signed languages or any?
No, sign language is a really tough one. The problem is not the nature of the sign, but the problem is the privacy concerns are just extreme. It's been really difficult. There are some banks, but no, yeah, it's really a problem.
Can students use it if they do not have a faculty sponsor?
They use, first of all the open databases are open, and you know, it's really easy to get you. This is, I know it seems like a barrier, but it's really easy. And if someone doesn't have a faculty sponsor, I mean, I guess I'll sponsor them, you know. Mostly, the reason we want the faculty sponsors, we think, you know, students have much more energy than their advisors, but we want to get those advisors somewhat involved too. you know, to make them aware, so that, you know, there's sort of the hysteresis of the whole thing. Yeah.
Yeah. There was a question that kind of disappeared, but just how consistent codes are across lab sites. If you want to speak to that, I can speak to that about AphasiaBank.
Oh, yeah, I mean, well, the coding system of chat is totally, everything is in chat. So that's nothing. There are no other databases in spoken language like that, where everything is in the same transcript format. Yeah.
Yeah, so we try and keep it consistent across lab sites. You know, reliability is done at the lab for the most part, you know, reliability of coding and whatnot.
Yeah, I mean, obviously, now when you talk about coding, there's a difference between transcription and annotation and coding. So I think the transcription is really, really tightly specified by chat. But if then people want to code additional things, you know, that's sort of project specific, and there are methods inside the CLAN programs for tracking that. But you know, we don't have a discipline. Everybody needs their own codes.
Yep.
Yeah.
I mean, we kind of make up a few of our own just because we're interested in.
Right, but those are add-ons once you have a basic transcript, yeah.
Right, exactly. Yeah, but I would say, you know.
And then to be clear, we really prefer to have transcript linked to media. So older data don't have that, but we've really been pushing to make it so that the media, either audio or video, and a lot is video, so yeah.
And I didn't show it just for PHI reasons, but yeah, when you go to the browsable database, everything is locked. The transcript is actually locked to the video, which I think is fantastic. So you can, you know, go and click anywhere in the transcript and it goes automatically to the video and you hear that person, which I think is a really great thing that TalkBank does.
And that's also true for the TalkBank DB. So once you get this output, it still has the link of every utterance that matched back to the transcript, and you can, you know, just go right back. Right. Yeah. Any other questions?
Sorry, my dog is barking, just ignore it.
I think Bree got most of those questions from the Google Doc out. There was one, are there any samples of children with TBI or aphasia?
Children don't get aphasia. There's no such thing. Children with TBI, but we have a lot of TBI. No, we don't. Thank God, there's very little children.
I don't think I know of any.
But we do have some young people with TBI, down to, I believe, 12. But really, TBI typically is motorcycle accidents. It starts around 30. Yeah.
Okay, so thanks, guys. I'm sure we have plenty more to discuss, but I'd like to get.
Yeah, we'll move on.
Yeah, and I really appreciate you both doing this. I'm going to try what we did in the last session. So I will very soon unmute everybody, and then hopefully we can clap to thank these people that, again, jumped in at the last minute to present this awesome stuff. So, okay, get ready, guys. Okay, 3, 2, 1. Clap. Okay, looks like my dog was not as upset about this as the last time, so that's good. He's learning. Now I'd like to welcome Josh Faskowitz. He's a PhD student in neuroscience and psychology at Indiana University, and he does work in the computational cognitive neuroscience lab. He's got a lot of experience in using completely publicly accessible online neuroimaging data sets. And again, as we mentioned, online data collection is good, but just getting data that's already been collected is better. So this is something that, you know, modulo all sorts of other concerns. But anyways, I'd just like to welcome Josh, and if you can take it away, that would be great.
Cool, yeah, hey, I'm Josh, Josh Faskowitz. I'm a student here in Indiana, and I'd just like to thank you guys for the opportunity to share my knowledge here, all this data that I've looked at over my years in grad school. So let me set up the screen share thing here. I'll share my screen. Share. Okay. Ooh, this, and then let me try to. Okay. I can't. Okay. Present. Okay. Cool. So. Yeah, I kind of just made a whirlwind of a tour of some freely available data across the web. Just some background about me. I do brain networks. I look at structural brain networks, functional brain networks. We merge structure and functional brain networks. And over the course of my studies, there's so much free data online. Fortunately, I haven't had to collect a lot of data that consists of a large chunk of what I've done. So these are examples of some of the data that I've used, and I'll describe to you some of this stuff. And so I guess I would consider myself a research parasite. This is like a phrase that has come up recently about people who use other people's data and make new hypotheses about it. And we totally depend on other people collecting the data. And it came up as a negative thing to begin with, I think in a New England Journal of Medicine commentary, but we've since owned it, our community of research parasites. So yeah, so I'm very appreciative of everyone collecting data, MRI data specifically. And so I'm not affiliated with any of these resources that I'm about to show you. I'm just going to give you my user level experience and some practical experiences with this data. So one of the places that has a ton of data is Figshare. You can Google it pretty easy, figshare.com. So sometimes when I'm bored, I just Google MRI T1, resting state, MP rage, just some key terms. And also when you're publishing your own study, an MRI study, and they ask you to share data, this is a great resource to put data. It's they give you a pretty large amount. I don't know exactly the numbers of gigabytes that they give you, but they allow you to put a lot of data here. And so there's been some stuff written about open data, Russell Poldrack being one of the big proponents in MRI about sharing data. So we're going to go through this pyramid a little bit. So at the top of the pyramid is the what he calls potential for reuse. So at the top of the pyramid is like results, someone's analysis. Neurosynth is a super cool website. I'm hoping I can actually preview some of this stuff. So if I click here, yeah, and I'm still screen sharing. Neurosynth is a great web. If you just want, let's say you want to do an analysis of just a a meta-analysis of of results across neuroimaging studies. Neurosynth has collated coordinates in a machine learning manner. And they have these maps of activation for you. And you can actually, so if I just clicked the link for language, but you can use any search term here, and you can actually download this map for your own purposes. So you can I mean, if you're interested in default network, for example, this is a meta-analytic map compiled for you, and you can download it here. So it's a resource for free. Brain Map is a similar one. This is it has peak coordinates, so you can put your you can extract peak coordinates from this one. Neural Vault is a place where you can download and upload maps, unthresholded statistical maps from your studies. So it's kind of like Neurosynth, but this is not just the results of the study, but not just the significant clusters, let's say, but the whole unthresholded map. Okay, so one of the big, if you're a neuroimaging researcher and you just want data available, Human Connectome Project is like a one-stop shop to get a bunch of data. So Human Connectome Project, the main study was over 1,000 subjects at a 3T scanner at WashU in St. Louis. And they since collected around 180 more subjects up in Minnesota with a 7T scanner. And they really went extensive in their imaging protocol. So this huge project, this part of it wrapped up, the 1,000 subjects, they're pretty young. I think it's like around 25 to 35. There's a twin structure here. It's a really big data set on the level of terabytes, the raw data. You can access it through, so this data set, to get access, you have to sign up. So it's not a point and click and then you get the data automatically. You have to do a little waiver, but it's pretty easy I signed up for it a while ago, and it took a week or two, but and I had to be underneath a PI, of course. But once you get access, you have access to a pretty high resolution T1 weighted image and T2 weighted image at 0.7 millimeter isotropic voxels. The resting state, it's sampled at a TR of 0.7 seconds. So, and that's for an hour. They have 4 15-minute sessions. So that's pretty cool. You're into functional network reconstruction, such as like I'm interested in it. They have a lot. They put people through a number of tasks. The tasks aren't so deep. Meaning, I would say that, I mean, they're not as, they kind of were just doing a wide survey of tasks, in my opinion. So they have working memory, gambling, motor, language, but they still, like the resting state functional scans, it's pretty highly sampled, so it's pretty good quality data. And then diffusion imaging, so you can run your favorite tractography with these data. It's a pretty good diffusion acquisition. One of the coolest parts about this project is that they pre-processed the data for you with their minimal pre-processing protocol. So you don't have to worry about, they did a lot of technical advancement, and you can just download the results of their pre-processed data. So they've already gotten for you the movement parameters, let's say, and they've already normalized it to M&I space. They actually even created their own proprietary format called SIFTY, which has both the surface data projected to a cortical surface in a standard space and the subcortical structures. And let's just say you're not even interested in all the data, subject level data. They actually have pre-processed group average, Cohen's deactivation maps for their tasks. So if you just want to look at a map of activation and kind of just do your work from home during this quarantine stage, and you can also download that. So that's pretty useful, like they did in this paper, for example. And the cool thing, so HCP is such a pervasive, I mean, we probably heard of it, a lot of us have. There's a lot of people that have released their versions of the data. So here's some examples I've included links to where people have processed 1,000 structural brain networks, for example. You can download the 70 tractography data, time series at the bottom here. So there's a lot of options. Now, so HCP, you have to sign a waiver. INDI, the International Neuroimaging Data Sharing Initiative, is just point and click for most of the data. You just go to this link right here, and you can get access to a lot of different data studies. I'd say that this is, in my opinion, one of the older data sharing initiatives in MRI. So there's a lot of different studies. I would estimate over 30 or 50. And for most of it, you just go and click the data and you start downloading to your computer. So it'll take a while. Some of the data sets you need to register with NITRIC. NITRIC is pretty easy. It's a neuroimaging and informatics resource. You just have to sign up there, so just that they have your email. But here's a view of some of the samples that they have. I mean, I could just click, but you could see that they tell you, I mean, there's 200 scans here, 28 scans here, 25 scans here. There's a lot. So I don't think anyone is going to struggle for general data. Again, it's not so specific, but if you just want to play around with some data processing, INDI is a great resource to look at. I have some highlights from INDI that I like. So NKI Enhanced is a particularly good data set. In my opinion, it's a lifespan data set. So it has data from 10 to 80 years old. And they also have deep phenotypic information. The phenotypic information you will have to apply for to get a waiver, to get all these tasks and the physical measurements. But once you get that, it's pretty deep. I didn't mention that HCP also has pretty deep phenotypic information. ABIDE is a large autism data set, 1,000 combined autism and typical controls, mostly resting state data. ADHD. SLIM is a longitudinal data set from China. MPI is another data set, the mind-brain-body data set from Leipzig. This also has pretty deep phenotypic information, and there's a Nature Scientific Data article. Atlas is an openly available data set with lesion segmentations from USC, the Southern California USC. So that's cool to check out if you're into some lesion segmentations already done, if you want to test your new lesion segmentation software. How about some clinical data? Well, so these ones you definitely have ADNI and PPMI, Alzheimer's and Parkinson's data sets. They're huge, over 1,000 and over 500 each. These you do have to go through a little bit of a lengthy application for, but they're out there. Prevent AD, they recently wrote a preprint, and I don't really know much about it, but it's available. It's available. And Abide, so they even have more autism data. Movie data, so CanCan is 600 people, over 600 people from Cambridge, collected at Cambridge across the lifespan, and they have rest and movie fMRI, and a good amount of behavioral data. This one you have to apply for as well, but it was pretty easy. They just took a while to get back. Like signing the form, it was didn't take much, but it took them a while to respond. Study Forest is around 20 subjects, but they watched Forrest Gump in the scanner. So that's pretty cool if you wanna, and they also, this data set in particular, they did a great job at annotating the Forrest Gump movie. I think there's a group from Germany, so they have all these annotations in German of like what was shown at the time. And then Jim Haxby at Dartmouth has openly available fMRI data from someone watching Raiders of the Lost Ark. The Healthy Brain Network Serial Scanning Initiative, also 10 people that watched Raiders of the Lost Ark, plus 10 sessions of resting state, and also like watching movie trailers. Healthy Brain Network, so the other one was like a test phase for this larger study, and this has a lot of children in it, and they watched a movie. I think they watched Cloudy with a Chance of Meatballs. I don't know much about the EEG, but they had that available here too. This one, I'm not sure if it's a point and click and download or if it's a waiver. And then deep data on single subjects. So My Connectome is Russell Poldrack's data set where he scanned himself 60 times within a year. And he actually has the serum data on his blood. And he has some tasks, resting state. So it's one person, but really densely sampled if you wanna, and they actually have pre-processed data through fMRI prep available for this one. Simon is a guy who got scanned at many different locations around Canada, I think. This is Todd Constable's data set, where he scanned 30 sessions within 10 months. And he watched a couple movies, he did resting state. That's available to download. And Midnight Scan Club, this has been a data set that was recently used. 10 people, 10 sessions, and it's pretty high quality data. The Grattan Lab actually has, at this link in the slide, they have data available to download, like already time series available to download for this data set. So that's helpful too if you don't even want to do the pre-processing. And then we even have non-human, so monkey data available from the group in New York, is available. And then Allen Brain Atlas, this is not MRI, well, there's some of that, but this is a whole, Allen Brain Atlas is a great resource for mouse brain connectivity. OpenNeuro, so if this is all overwhelming, OpenNeuro might be a little bit more overwhelming. I just want to click here really quickly about OpenNeuro. I mean, overwhelming in a good way. OpenNeuro is a place where people deposit their data sets. And they have a philosophy of storing their data in the BIDS format, meaning that it's just organized really well for you. But you can click a data set here. So I just click this random data set, and then it takes a while to load, but you'll see that these data are available. You can actually, so here's the study. Usually there's a README, some acknowledgements if you reuse the data. They ask you to cite here. And then I'm just on the right side here, clicking through each subject, and you can just see the data that they have, anatomical data, functional data, and you can download these in bulk too. So it's really helpful. You can browse this website. You can see if you have people with similar hypotheses, and you can browse their data here. So that's really useful. I just have some pictures from it. Finally, so this is an initiative from the Pistilli lab here at IU. I can't get too in depth into it because it does a lot, but this BrainLife.io platform, you can add data in the cloud via this platform. I just recommend going to the link and looking at what they have to say here. But basically what you can do is they have the computer set up for you. You just click the kind of processing. They do FreeSurfer, they do fMRI prep at this website. So it's pretty cool. I'd recommend checking out the resources. And then finally, since I'm a networks guy, I actually have, if you go to my GitHub, I have compiled a whole page of openly available network data for you to play around with. So this is just at my GitHub. I have non-human animals, I have human animals, network data, so that's available. So, and then finally, if you want some help, go to neurostars.com. I really like this resource. So that's it from me. You can hit me up on Twitter, or I'm gonna share this PowerPoint with all the links in it. But in this time where we're all at home, there's definitely a lot of data to play around with. We're not alone here. We have a good community of very generous people, and I thank them as a data parasite myself.
Thanks, Josh. That's fantastic. And I just want to particularly thank Joshua for doing this, because I think literally he was asked maybe last night at like 5 or 6 p.m. to pull this together. So thank you for doing it at the last minute. That fit perfectly. So as Josh said, he'll send the PowerPoint so we can upload that, and you can access all those links and resources. So I'll just ask a couple questions, if you don't mind, from the Google Doc. Mm-hmm. So the first one is asking for the resources. So yeah, we'll make sure that those get linked. And then the second question is more general about, so this person said, I'm not familiar with using big data or data sharing from other labs. From your experience, how is publishing with it? Do reviewers criticize it? Is it pretty welcome in the literature?
Sorry, publishing the...
Yeah, so what's, if you were to compare publishing a controlled experiment versus data that you got from somebody else, how does that fit with reviewers and journals? Is that...
Yeah. So I try to do, that's a great question. In terms of, because I'm not the one, me personally, since I'm not the one collecting the data, I actually do feel compelled to share the derivatives of my study. For example, I had published on that NKI data set, and I actually uploaded the networks that I generated, so that I'm doing my part too in sharing the data. This was also, though, there's some trickiness of, in any of these situations, you should consult like the documentation from the original data source and make sure you can share derivatives. For the NKI, it's one of these data sets where you can kind of point and click, so it's a pretty open data sharing agreement that they have, let's say. But it would be a case-by-case basis with other data sets. For example, ADNI, which is like clinical Alzheimer's data. You would have to check in with their documentation to make sure you can share the derivatives. But other than that, yeah, I mean, you didn't collect the data, but you're still using it. Obviously, you shouldn't share the raw data on someone else's behalf. But I would say it's a good gesture to share what you've done with the data that's unique on your end. I think you're muted.
Sorry about that. Yeah. So I think that another part of that question is really just, is it harder to publish using borrowed data or shared data than your own data?
Um, hmm. This is just the perspective of me as a student. I think it's harder to generate hypotheses. I mean, in terms of you have the restriction of using data already collected. So your hypotheses might not be able to be as you might not be able to tailor the data to answer the exact question that you want. So in that regard, maybe it could be harder. However, I mean, from my perspective as a grad student, I don't think it should be looked down upon or anything from a journal editor, let's say, of they're be reluctant to publish it just because it's not your own collected data. I haven't heard any stories about that or anything, but hard to know. I'm not an editor or anything.
Right, right. I guess we should all make sure to browbeat people, to make sure that they're okay with doing this sort of thing, because especially now, right, it's not like we have options anyway, but it's also really expensive and can be difficult to collect. You don't have memorized data where you are. Another question that I just got from the chat was, I'd like to know to which extent data are raw data,
Right, yeah.
So like, I guess the question is really about like, if data have already been processed, how hard is that to work with?
Yeah. Well, so most of the data that I shared here, they're all nifty format. There's no DICOM, so it's a little bit above raw. But like for HCP, for example, that's pre-processed, and they have raw. But I wouldn't actually recommend downloading the raw because it's terabytes of data. The OpenNeuro, that huge website with all the studies, I'd say they have over 100 studies on there. That's nifty level data. And that's not pre-processed. So that is, but you also should do, whenever you're working in the situation, you should do the legwork to make sure it is what you think it is. I say this because, for example, on some OpenNeuro datasets, some individual datasets, they've skull stripped the T1, for example. And so you wouldn't want to apply SPM normalized to a certain atlas if it was brain on or skull on or skull off. So that's kind of the things you have to just check out with the documentation. So all these resources should be well documented. And maybe from my perspective, if it's not well documented, it's probably not worth using. So because then you don't know if they've if they've nuisance regressed the data already. You don't want to redo that, for example. So stuff like that. I mean, it's on a case-by-case basis, obviously, but generally the resources I have here, they're well documented. They'll tell you what's raw, what's pre-processed, but most of it is nifty format, unprocessed.
Cool. So another quick question is there a convention for citation of data? Like, do you just cite the paper? Is there some other thing you have to do?
Yeah. That's a fantastic question. OpenNeuro, again, I keep on going back to this. They ask that you cite OpenNeuro. And I think they have I think they force everyone. I think if you're going to post your data on OpenNeuro, you have to accept their Creative Commons license.
Yeah, I was looking at it last night, and that's what they said. They make sure that everything is going to be Creative Commons.
Yeah. And but ADNI, for example, they have when you sign up for ADNI, the Alzheimer's Data Neuroimaging Initiative, you have to you actually in some cases had to add them as a last author. So that's a totally different level of acknowledgement.
That's a pretty big jump there.
Yeah, that's a pretty darn big jump. So, but I would say it's just if there's no explicit, if there's no agreement, if it's really open, for example, indie, I think it's still a good gesture to put it in acknowledgements when you're publishing, just so that your readers are aware, and just so that they at least have some sort of metrics. in terms of when people Google the NKI data set or PubMed or something that shows up. But there is no standardized. I think people are pushing for a standardization of assigning credit to data. I don't think there's a consensus. But I would say that the best practice is to, when in doubt, just put it in the acknowledgements at minimum.
Yeah, that seems like a great option. So another question here, this may be a little off topic, but has using shared data opened up collaborations with other researchers or sites? Or do you just typically work independently once the data is available, you just download it? Or do you more work actively with those researchers? Looks like either me or Josh is frozen right now. And I'm not sure what it is. So I'm just asking if people can hear me or if they, okay. So it looks like Josh is frozen. So is Josh frozen? Please indicate. Okay, all right. Well, you know, Josh may or may not be able to hear us, but this is being recorded. So, you know, he'll be able to go back to that Google Doc and answer your questions on that, hopefully. But at any rate, whether Josh can hear us now or whether he will come back and view this on his own time, I'd like to do, again, the applause. So again, I'll do the same thing. I'll count down, and then please just give your applause to Josh. Thank you for getting this together at the last minute and really helping us out. So 3, 2, 1. Awesome. Okay, so we're at 2 o'clock, but I would like to just stick around if people are okay with that for just a few more minutes. And just basically the idea was, you know, we've talked about a lot of interesting things, publicly available data sets, methods for collecting data online. But again, maybe the easiest way to go about this is someone published a paper, you could realize you read that paper and you're like, I could probably use that data to do X, and you can just ask them for the data. And I think it's often the case that people are willing to share that. So basically, I just wanted to bring Bree back. And if there's anyone else that has a lot of experience with this, please just go ahead and indicate, you know, in the chat if you are interested in sort of talking about this. But I thought that Bree would be good because Bree has a lot of experience in asking people for data. So again, like I said, if you are interested in chiming in here, please just go ahead and indicate that in the chat. I'm gonna actually, looks like Josh is like thawed, so I might try to bring him back just in case.
Yeah, hey, I'm sorry about that.
No worries. But maybe you want to chime in on this topic too. I'm not sure if you're familiar with directly asking people for data, but at any rate, I think this might be a good place to get a lot of questions. So I don't know, I mean, from my experience personally in graduate school, I mean, I started off and I probably could have done this, but I think I was very shy and did not want to just ask somebody. I felt too brazen to do that, which I didn't do, but I think in retrospect, it wouldn't have been a big deal to do that. So that was my experience. I don't know if other people have that sort of experience, but if you have questions about that or concerns, please just go ahead and go to the Google Doc and just type your questions or comments if you want to add comments. And again, like I said, if you have some experience, please chime in here. But I don't know, maybe Bree, could you maybe comment on this a little bit?
Yeah, I think this is really tricky when you work in clinical data like I do. A lot of, it's still kind of gray territory whether or not lesioned brains constitute protected health information. So it depends kind of where you are. I know in the UK, so I did my PhD at Cambridge, so I was based in the UK. Their ethics basically state that you can't share any brain that's not quote typical because it does contain PHI data. Here in the US, the IRBs kind of vary depending where you are. They're more stringent at a hospital or a medical university versus at not a medical university. But something that typically has worked for me is just asking for standardized lesion masks. So masks that are already standardized to something like the M&I space, to which you can then do some analysis on. And most people are many people are free to do that, but as someone who collects clinical data, it is a lot of money and time to get this information, and it is difficult to share. But I think it's always worth an ask, but my caveat is to let people know why you want it, exactly what analyses you want to do, and in the first email to say, let's work out authorship criteria, because I think that puts forward, I respect that you collected this data, I want to give you whatever you think that you need in terms of getting people to give you credit, and that typically has worked out the best for me, I would say.
Right, and that's a great point that you make, right? I think if you are offering authorship, I think pretty much everyone is going to want another paper on their CV and citations of their pre-existing paper. So I don't think this is something that people are I mean, I can imagine situations. I've known some PIs that are more protective of data, but I think once they've published a paper already on it, for example, then I think it opens up a lot more flexibility and openness to sharing data. I'm trying to think of, I feel like I have some more thoughts on this. Does anyone else want to chime in here? Bree, you can go ahead if you want, or someone else has a question, or like I said, if anyone has experience with this, please feel free to add something if you'd like. Someone asked a question on the Google Doc with regard to authorship. Would the data collector go last or just somewhere in the middle?
For me, it's kind of dependent on the preference. I think in neuroscience, typically the most senior author is either first or last. So a lot of times the people sharing are fine with being in the middle because they collected the data, and according to like APA agreement, data collection alone doesn't necessarily constitute senior authorship. It kind of depends on what authorship criteria you're honoring. But I try and be very forthright with that and saying, this is what we typically follow in terms of APA. Do you want to talk about it? But typically I see them, at least in neuro, falling in the middle. Josh, I don't know if you have had a different experience. I know you said Adney likes to be senior author, but I don't know if you've had any other different experience with that.
So here at IU, I've actually helped pre-process data for other labs. And so as a data pre-processor, where I'd actually take the raw data and pre-process it, I've been a middle author for that contribution. But never have I been as a data collector. The only thing that I can think about is at USC in California, they have Paul Thompson's Imaging Genetics Center. They have the Enigma project. And there they collect data across, they collect thousands of subjects. So they have a 200-person author list, and everyone who's collected data and done the basic pre-processing to aggregate the data is in that middle 200. And then there's a block of senior authors at the end who are, so maybe like 3 senior authors, and they're in a little non-alphabet, that middle block is alphabetized. Then the last block is not alphabetized, and then the first block of people who did the first draft is they're there in a first author block. So that's my only experience with this, is in terms of that's just a bunch of data providers, and not just though. I hesitate. I mean, they're providing an important part of the Enigma project in those instances.
Right. And I think an interesting point to bring up here is if your research is federally funded, there is in some sense an obligation to disseminate your research and your data in some sense, right? So I think this is something that for us to think about. It's something I've never really done. I've never publicly posted data. At the same point, is there some obligation that I have because my research is NIH funded, to make sure that that's available so that the maximum, gain can be had from that data that was collected. So it's I think an interesting point to make, Does anyone else have any questions, comments, concerns? Anyone want to just like go back into Finding Five and just like, practice? I guess not. So if that's it, then I just really want to thank everyone who participated that's still here. Bree, thank you for just responding to my email yesterday, and Josh, like I said, for coming online. And then Noah from earlier, that was a really amazing presentation, and everyone else that partook as well. So we're going to make sure that these recordings are posted. I'm not sure exactly when and how that's going to be done, but I'll make sure to disseminate that information. It may be the case that I post all these recordings on the CUNY conference website, maybe along with the recordings from the conference itself. That might be a good solution. But we'll be, however you found out about it for today, then you're going to find out about it for the posted videos. Please feel free to share those. I think that's pretty much it. I don't know how you're supposed to end these things. I don't know, but anyway, I'm just gonna say goodbye, good night, good luck, live long and prosper. And you know, like I said, feel free. You can ask us for data. I know that I'm willing to share anything that I've published. If I can still find the data, I'm perfectly happy to share that with you as well. So, all right, thanks guys. Goodbye. And thanks to the CUNY organizers for making this happen, really, because if they hadn't followed through and actually hold CUNY, then this workshop would not have happened either. Okay, bye-bye. I guess I'll stop recording.